15. I am turning off Z-bias. 10.
Good morning. We have the bottom in sight. We are commencing with dive number 6.
I'm putting in auto heading.
The windows to the deep.
All right, when's coming to an easy
2018 expedition.
Copy.
Delta 15.
We are at a water depth of almost 780 meters.
Okay, backing.
Our current location is about 170 kilometers due east of Jacksonville.
You know the drill when you catch me in your fisheye.
And we are just east of the primary axis of the Gulf Stream.
Should I go port starboard?
And we'll let the pilots get the ROV settled.
Going up the ridge.
And we'll do a little more introduction.
So yeah, starboard.
So stand by.
Backing pretty hard, mate. Not making a lot of progress.
I got you in Titan.
Yeah, so most
you
of
got me in your fisheye yet?
Should be coming front back.
Okay. Hard to say.
Okay. I think you probably have room to spin around and catch me. If you want to try.
Okay. Come to starboard. I got you, fisheye.
Copy. Seeing your light pool swing around. Alright, you should be pretty much lined up with me. There I am. Great. Pushing down.
Come down with you.
Adjusting my Z-bias.
All right, winch will stop, unless you want me to come down further.
No, that's good. Just coming back towards you. Sort of line up. Let's see. Do you want to just catch me due north?
Yeah.
Go ahead, Nav.
I'm just wondering if you finished the heading plate?
Yes, I'm done.
Copy that.
Thanks.
So we just hit bottom.
Okay.
I just wanted to brief you on the bailout. It's going to be south at the moment.
Okay, so we're going to do a white balance now that we're down here.
Protocol, confirm that the rotation, center rotation is at A frame.
Correct. Center rotation is at A frame, and I put the bailout at south.
Copy that. So soon I'm going to call you for the move. We're going to be moving probably.
Okay, with me taking your hydraulics?
Okay.
Yeah, okay, I'll put you at 5.5.
All right, thank you. I am ready, yeah.
Okay, right, I'll call you back.
Okay.
Okay, there's 5.5.
Pilot and co-pilot, let me know if you want me to change any settings on the sonars.
No, I think they're okay for now, thanks.
Copy.
Okay, video. Let me square it up here. Okay, video, how does that look? I can bring it left a smidge if you'd like.
Yes, please.
Yeah, I'm working on it. Trying to do some small adjustments here.
Camera's at 7.3 now.
Yeah, understand.
It's the wrong place.
There we go.
It's good.
Just confused.
Would you like me to rotate it down to smidge, Roland, or is that okay?
No, that's close enough.
That's close enough. Aye.
Capping the camera, checking the blacks.
It's pretty stationary. Firing 45 left, 40 back, or 30 back.
Tether's pretty tight.
Oh. Backing up.
I have your mini Z's.
Copy.
Looks pretty good. Rotate for
For our
HDQ.
viewing
that.
audience, what
Keep it
you're
rolling.
seeing is our color balance test. Now you see the manipulator arm. We were zoomed in. And in the distance, you see the sea floor.
And it looks like we have a lot of coral rubble like we saw yesterday on the seafloor here.
Definitely.
How's that rolling?
It's good.
Copy. Does it look similar to bottom as yesterday?
You'll hear a lot about that today if you stick with us throughout the dive.
Yep.
And realize its importance to this environment.
That looks like a midwater fish there. And a pancake urchin right in the middle of the screen. We saw quite a few of those yesterday. Ariosoma is the genus. And those have poisonous sacs at the end of their spine. So if you ever come across one, you want to make sure you're wearing gloves and try to pick them up.
You're not likely to find one on the beach or anything.
No.
We are at a depth of over 800 meters, located
Video's clear with
due east of
HDQ.
Jacksonville, Florida, about 170
Thank you, video.
kilometers.
Still the arm.
If you're just joining us, this is the northern edge or northern end of the feature called the Stetson Mesa. Very flat-lying area, completely pockmarked with these biogenic mound-like features. Well, they are mounds. And we explored one yesterday in great detail, actually a series of 3. And today we are far north of where we were yesterday and looking at an even larger mound. So we're expecting to see something very similar and maybe even more dramatic as these features reach upward into the Gulf Stream that provides current flow that reduces sedimentation so that they don't have to get buried all the time. And it also provides nutrients and food, especially for these filter feeders and suspension feeders.
Okay, looks like
Yeah, the corals that live at these depths are relying on food from the water column. They don't have the zooxanthellae or the microscopic algae that the shallow water corals have that people are probably more familiar with. So being up off the sediment and in high water flow delivers more food particles to them. So that's the desired habitat that they try to reach. So the high parts of these mounds are the prime real estate. And we'll probably see, as we did yesterday, the cover of live coral increase as we get to the top of these mounds. Looks like we have a lot of...
We're here on bottom if you guys are ready.
Yeah, I was just noticing these octocorals here. We collected... two species or two individuals yesterday. We were unsure if they were Unicella or Swiftia. Once we got them on the ship, they looked more like Swiftia, both of them. So, of course, this was to an untrained eye. I'm not an octocoral taxonomist, but this sure does look like that species that we collected yesterday, and it looks more abundant here. Yesterday, we just saw a few of those along the whole dive track, but it looks like there are dozens here already. That's really exciting. Oh, and the first synaphobranchid eel, cutthroat eel.
We saw many of them yesterday, so they definitely like the flow of the current as well in this habitat.
Yep, and they are typical of the Lophelia and Analopsamia, which is the other scleractinian coral that we saw a lot of yesterday. They're very typical of those habitats, so expected and right on cue.
Box in all the way.
Like and the scleractinian corals are the ones that make a calcium carbonate skeleton, and so they're a little more sturdy, maybe. They're not the soft corals, and when they die, their hard part, that calcium carbonate skeleton matter, remains on the seafloor long after the organic polyp has disintegrated or degraded or been eaten by something. And so what you're looking at is a field of rubble of those skeletal pieces. And even under the sediment areas, the flat portions, you see all those. Those aren't worms or worm tubes. Those are skeletal pieces, and we call that coral rubble. And the dark brown that is coating all these pieces is iron oxides or iron and manganese oxides that precipitate out of the seawater itself. The living corals, though, are these the bright white stems
settling in here.
And we'll be zooming in so you can see the hard part as opposed to the soft coral tissue.
Crinoid coiling up very close to your lower right.
Yeah, and both of those scleractinian corals we've been discussing, Lophelia and Analopsamia, are white when they're alive. And from far away, when the polyps are extended and you can't see inside to the calyx that the polyp sits in, it's difficult to tell the species apart. So actually zooming in on the dead coral framework is a better way to determine the species often.
And this pancake urchin is a cousin to sea stars and brittle stars, and in fact surrounding this organism you'll see the skeletal material of the dead coral branches, but all intertwined. You can see in the lower right, you'll see arms, tentacles, and those are the brittle stars that are very common around here.
Ophiuroids, and they're both in the class Echinodermata. Oh, phylum.
Phylum.
Echinodermata.
Rotate left.
Meaning spiny-skinned.
As are sea stars and crinoids and even sea cucumbers.
And we have a chance to see all of those today. I think we saw everything except a sea cucumber yesterday.
That's correct. We did not see a sea cucumber. And we only saw one sea star, which previous dives farther out on the continental margin, we've seen several different species of sea stars. So we were surprised to only see one yesterday.
Sure.
Beautiful, bright yellow one, though. So, oh, and look, there's a sea star right in the foreground on the left, on cue.
So the organism in front of the sea star is Duva florida, which is actually an alcyonarian octocoral. So that's a soft coral. So it doesn't have the hard parts or spicules.
Okay.
Yeah, that's the Duva, the pinkish
organism. The pinkish one, yes.
Yep, that we're zooming in on now. So that one can kind of totally deflate and retract.
Towards him a little bit, you probably could.
We saw one that was in that state yesterday.
I think it's a good start, 20 meters north.
Sclerates, not spicules. Sorry, spicules are sponges. I knew that was incorrect. Oh, and there's a little squat lobster right at the base on the left, lower left, under the Duva. Probably a Galatheid squat lobster.
Yeah, that shouldn't even put you right over it, so I'll just call it.
I don't see the squat lobster. Yeah, go ahead.
Go ahead, Nav.
Full screen for the corals.
You can see a little crinoid, another echinoderm that was just in the center of the screen. And there are several of these maybe Swiftia octocorals, the big white octocorals that you see.
Okay.
Actually, they're not that big because the lasers, the red dots that you see, are 10 centimeters apart. So although they look big on your screen potentially, we have to keep reminding ourselves that they're actually, those are 10 centimeters apart. And so a lot of these organisms are actually quite small.
Did you want lasers back on?
No, I wanted lights back on.
Oh, lights.
That sea star has a lot of arms.
2, 4, 6, 8 arms,
Bug lights
maybe?
are still on.
Maybe.
Okay.
Yeah, I just got a
Some
little
of them are
bit farther
hidden,
away
but
trying to get in here.
It's an octostar.
Yeah.
Back in.
They usually have 5 arms.
Ready?
Yep.
Now you can see why we call them echinoderms, the spiny skin.
Wow, that's a great shot.
It is great. It's reaching underneath the coral rubble. You can also see that iron staining on the dead branches of the coral. And at the very tips of the arms or the rays of the sea star, you can see the little red spots. Those are the eye spots. They're sensory organs. And the little tube feet that extend from the ends, but also certainly throughout the underside of the sea star provides mobilization, and a little brittle star on the upper right of your screen.
Video's clear.
Both echinoderms, but in different classes. Oh, that was a nice little menagerie, just for our very first view of the dive.
And that's something that we'll probably continue to see that we saw yesterday is when you start zooming in and looking at the framework of this dead coral, it creates a habitat for a plethora of other organisms. And so the more we look, the more we see. And as Leslie said earlier, that's a very important part of this habitat is the
those lights may or may not bring out
structure that is created by these corals, even when the corals are dead, provides homes for lots and lots of organisms. And you can see that this is a rather fragile structure also. If this were swept over by a big net or something like that, it would remove that structure and all of those organisms within it.
We're also seeing a lot of sponges. We'll come across them throughout the dive, different morphologies of their
Orbit,
shape,
pretty good.
different characteristics. The one moving into the center of the
Yeah.
screen
I
now,
think it's a temperature.
we've been referring to as a satellite dish.
Small model.
Although
Has
I'm
to have
not
a
sure
small
that this is
amount.
the
Maybe
same
a,
species
maybe a
as
different
the deeper.
species.
You know,
I mean the
deeper satellite.
morphology. Yeah.
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 This one looks a
Just
little
the
thinner,
coolest section
and
is that glass, I
You
guess.
see a few of them a little brighter white in the screen coming in on the left. And this coral rubble that defines the floor here, you can see that there's sediment lying in between it.
Left, it's got some
We
nice,
kind of think of
the
this as
old
almost
coral right behind
armoring
it.
the sand
Come
of
in
the
partial.
landscape here and
I love
holding
that
in
look.
place the sediments that have deposited. The currents are very swift here, as you might think, in the Gulf Stream, so the velocity of current prevents the very finest materials from settling out. It's too much velocity. So we'll try to get a sediment sample later on.
Oops, sorry, video.
It's always tricky, so we don't always
Myself
get
showing
one.
different in the current.
But
My
it's likely
offset had to
comprised of larger grains, and those grains are
Smooth.
almost
It looks like it's
assuredly going
smooth
to
up
be
ahead
the
of you.
shell remains of microorganisms that live in the sea surface, and when they die, their shells eventually settle to the sea floor.
And it looks like we have 3 octocoral species right in this view here. We have the yellow one left, the one that is Swiftia or something similar in the middle and right, and then
Yep.
possibly Plumerella or Caligorgia, which is the feather-like looking one on the bottom of the screen. Now that's a great start.
What about that single stalk there on the right? Is that a different one?
Yeah, they're actually, Roland just pointed out that there's another one on the right side, just inside the sponge.
The pinkish one.
Yeah, a pinkish one, and that could be a different species. There's, let's see, 68 species of octocoral known from the southeast U.S. coastal region, deep sea region. And I think it will take a little while of going through the video to determine how many we've seen on any one dive.
Going for a middle wide shot.
And Shirley Pomponi is adding that the sponge that we're looking at here, that's kind of a satellite dish looking
Tilt up into the center of the
sponge shape, could either be a demisponge or a hexactinellid glass sponge, and she's not sure. So again, sponges are another group that
Tilt up
are
into crinoids.
they require a lot more taxonomic work.
Excellent.
They're a very interesting and diverse group. You can see 2 little crinoids.
Crinoids, which are the other group of echinoderms. We've already seen all but the holothurians.
Weird.
Those are the sea cucumbers.
Salt.
And it was actually a sea cucumber that I found in a tide pool in Maine that started my fascination with marine life when I was quite young.
Lower bottom left.
Thought it was the strangest creature I'd ever
Cup
seen.
coral, maybe. Bottom of
Is
screen.
that a little
Yes,
cup
little
coral,
tiny
or
speck
what
in
is the
there.
pink?
I can't
Cup
tell.
coral or oh, it's an anemone.
Yeah, I
and
think it's
here's
an
another
anemone,
one in
yeah.
front of us here. Yeah.
Then we have more encrusting sponges, the
I lost track of it,
green
sorry.
and
There's one to meet you
brown,
right.
round-looking things, and possibly more of a glass sponge
Yeah,
that's
what's our
the
average speed
off-white,
needed
opaque
for today's dive?
looking,
Like the
kind of
last
left
one.
of center. And we have some baby stalked crinoids just above the anemone, the pink
Should be
anemone on the top right.
Easy for you to say.
It's not,
Skip down
It's
to the
not
bottom
for
of it.
me either.
especially with a cold.
Oh, yes. I
Sorry
don't have
if
that
I-
excuse.
Sorry if I sound extra funny today.
I just tell her she needs to get more
We could
sleep.
get another movie.
Which isn't very easy out here.
Okay.
There's just too much fun to be
Yeah.
had with looking
Yeah.
at all the
20
video
meters
and
good?
pictures that we
Yeah.
collect all day.
Okay.
The video's clear.
Oh, it's clear.
So Scott France is saying that the octocoral in the
I would just
foreground
watch Colin
here
movie.
that's
Great.
got thicker branches looks like Anthophila. And we collected
enough.
that in the Mid-Atlantic Canyons several years back.
Full wide.
Before we forget, maybe we should introduce the front row, especially because you're hearing already from our video lead.
And pilots.
Yes. Nav, would you like to start?
Sure. Or maybe the pilot can introduce everybody.
You could go ahead.
ROV bridge, move initiated.
Make sure you're on OKX.
Thank you, Bridge. So we are a group of engineers representing the Global Foundation for Ocean Exploration. Sitting as navigator, I am Fernando Aragon.
Sitting as pilot, Jeffrey Lanning.
Sitting co-pilot is Levi Unima.
On the far right, you got Roland Bryan.
And in the back, I'm Emily Narrow, and I'm in the video clipping chair.
Great, and they are a wonderful team. We will say this every time we introduce them, how great they are to work with, how talented they are, and it's really been a pleasure. And they have great eyes. They've been looking at these monitors a lot longer than we have, so they'll see things we're missing. We want to get that sea star over on the right before we pass over it.
Pink.
Oh, yeah, it's pink.
Stand by 1 second.
And there are two of us in the back row. I'm Leslie Sauter. I'm the geology lead for the expedition. I'm a marine geologist at the College of Charleston.
And I'm Cheryl Morrison from the U.S. Geological Survey Leetown Science Center. I'm a research geneticist, and on this cruise, I am the biology science lead. And I wonder if Chris Maw is tuning in yet this morning. He's our sea star expert, and often contributes great knowledge. And that sea star is sitting on a stylaster, which is actually a hydrozoan coral.
Maxim.
I believe this is a goniasterid, but I don't know for sure. I'm hoping someone on the chat room will correct or agree with me, since we don't have our echinoderm expert on the line.
It's a beauty.
It is. I think he wanted to be photographed.
Mm-hmm.
Video's clear.
Pilot's clear.
Thanks, Nolan Barrett. Definitely a goniasterid.
A beautiful glass sponge on the right.
Mm-hmm. You want to take a quick zoom at that? Or?
Yeah, and then we should probably take a quick zoom of the sponge to the left, the brown round one, which might be a geodia.
Let's try those extra lights.
Lights coming on.
And Nolan Barrett confirmed that that's a goniasterid sea star that we were just observing.
Come on in.
Beautiful glass sponges.
Yeah, we've seen a lot of these. They just pop out. I just think their white is so stunning. And that's likely a function of the silica in their sponge spicules, the composition. It's very glassy, which is, of course, why they call them glass sponges.
Seems to be a white claw in there.
Yeah.
And there's often a pair of shrimp that live. Yep, you can see just a ghost-like shrimp inside. There's often a pair of shrimp that will spend their whole lives within these glass sponges.
That one looked to have a bigger claw like a crab or something.
ROV bridge
There's
move
another
complete.
one there as well.
Copy, Bridge, move complete.
Yeah, they could be snapping shrimps that have an enlarged chela. I think that's the usual suspects that live in here, but they're so clear it's hard to see them. And
several crinoids on the right, taking advantage of whatever the glass sponge might expel, perhaps.
And Leslie and I have, and everybody who's touched our samples know about those spicules because they wind up in your fingers and they're like little needles.
Mm-hmm. And the sediments we've recovered are definitely showing them as prevalent as well. See something pink in there, but I can't tell if that's inside or outside or what it is.
We did see polychaete worm in one of these at the deeper site, and it was
There's
pinkish.
another claw down below.
Anything you can do to bring it out?
Yeah, I'm trying.
That's great.
Not much more I can do. Just get the
Okay.
blacks full crushed.
So Shirley Pomponi adds that these are the Euplectellidae glass sponges, possibly Raggedrella, but she's checking on that, and I think that Chris
Getting
Kelly
a glimpse of that guy.
thought it was
Shrimp.
that. Oh yeah, look at that view of the shrimp. That's amazing.
You see a claw on the one at the top of the screen as well, popping up periodically.
And then
Yeah,
you see
that's
his
a great one.
two reddish, pinkish eyes, and
Mm-hmm. Wonder what he's eating.
yeah, I wonder if he's cleaning some food off the inside of the sponge, or if he's filter feeding right along with the sponge.
Yeah, could you zoom out to the next waypoint?
All right.
There
If you
it is.
wanted to
Excellent.
see
Oh, you
that
got it
possible
selected.
demo
Okay.
sponge, we can mosey over there.
I'm on top of it.
Sled.
This camera is amazing, as
Hold
you
that shot
probably
for just
noticed.
a little bit. Real nice colors.
Say again?
Yeah, I was
Just
thinking
hold that shot.
Oh,
keep you
sorry.
going
That's all right. You've already moved. Keep going.
North.
Okay.
north until you're behind. and
I didn't
just
hear you
line
over
up.
the other chatting. Sorry.
Sorry, pilot.
No, not your guys' fault. You're good.
So, Cheryl, is this the one you're talking about?
Yeah,
Upper
that's
screen,
that's
middle?
the Geodia, potentially the Geodia genus that Shirley Pomponi is very interested in.
Geodia
Get right on top
pachyderma
of
is
the
what we're
container.
looking for specifically.
Say
Is
again?
that correct?
How close can you get to it?
Pachyderma meaning
Those
thick
sponges you were
skin.
just looking at would be under
Pachydermata,
my
thank
skin-
you.
skid if I got super close, but
Okay.
And
I
Shirley's
can
on the
get
line
close
with
and not
us.
land if you'd like me to try that.
Let's try this first.
Okay.
Yeah, I've got a fairly significant delay in the video, so it's probably, I'm trying to refresh this again, but so I don't see what you're looking at, if you're looking at a geodia right now. But I'll catch up.
Okay.
Is that a sponge or is it any kind of derm?
No, it looks like a sponge. It's got a lot of stuff attached to the outside, though. I think it's a sponge. Although we don't see the excurrent siphon at all.
Those spines aren't indicating some kind of urchin? I don't remember the pachydermata having spines like that.
Yeah.
It
I
almost
think
looks
that-
like a decorator urchin, or you know, where
Yeah.
they put the sediment
Try
over
to
them
get
as
it from a different
camouflage.
angle.
Yeah, try coming around. See what we can do with this current.
So some of the demo sponges can have fairly long spicules that make it a kind of a shaggy surface. Yeah, kind of like this one.
Are you seeing it yet, Shirley,
Yeah.
on your
Come on
screen?
again.
See, yeah, I don't know if what you're looking at right now is
Descriptive.
zooming in on this spherical structure, but to me that does look like it's a sponge with spicules protruding. It's not Geodia pachydermata though, so
Okay.
yeah, don't be concerned about collecting this one.
Okay.
Yeah, Fernando, I rotated slightly to port a little
But
bit.
interesting that
Better
it is
line
a
up
sponge.
uphill.
It was hard to even tell what it was.
That's all right.
It does kind of mimic an echinoderm.
Alright, I don't know if I'm going to be able to get it stable
Interesting.
from this angle. Sorry, Roland.
So you want the next move to
All right, so that's not one we're
340
going
or
to
a little bit? Okay.
look at
4-0.
collecting, I guess, unless we hear otherwise.
Don't call it in quite yet, but
Yeah, no, don't worry.
Extra lights are clear.
yeah, Jeff, I turned slightly to port just to line up uphill a little better.
Sounds good.
So we'll continue along upslope here. Looks like we have another one of the
Got my Z-bias tuned
pancake
for
urchins.
when I have my side in the current. But as soon as I put my back in my current, it just dive bombs you into the ground.
Yeah.
pancake urchins are Ariosoma. And a few more demo sponges, it looks like, and we continue to see this coral framework and rubble that coats the bottom of the seafloor here.
Is that moving? Oh, a crab, or oh, that's just coral.
Coral in front of a sponge.
Oh, no idea.
Okay, by calling move in if you want it,
Another
Fernando.
one of those spheres.
Copy that.
Yeah, another one.
Yeah.
Go ahead now.
Go partial if you want a quick glance.
New move.
Looks like we have another one of the sponges like the one we just looked at.
Okay, that's 20 meters, bearing 340, speed .2.
Video, can you brighten up Sirius HD just a little
Say
bit?
again,
Thanks.
Fernando. Yeah, it was ramping a little bit, but I think it's fine now. I'll let you know after this move.
Have more cutthroat eels in the distance here.
It's
We
the
really
purple
haven't seen
thing on top of that white stalk.
ROV now, Bridge,
much
move
live
initiated.
scleractinian stony corals yet.
Copy, Bridge. We started moving.
Copy that.
This here rolling you're talking about?
Yeah.
Yeah, you can come in a little bit. Just grab a glance, see if it's something we want to take a longer time to look at. I think we've seen that before.
We have a blue sponge. We had a little bit of that on the rock that we collected yesterday, the geo sample. Oop, and here's a good view of a cutthroat eel.
Alright, it sounds like they know what that is. Keep moseying. You're alright with that video?
Got a little bit of time.
So Chris Kelly was asking how deep the Lophelia and Analopsamia bioherms go down to, and is this close to their maximum
Get the one
depth?
behind there, maybe.
And
Give us a
I
slight
would say
tilt up. I thought I
this
saw
is
a
pretty
larger
close
fish
to their
back there,
maximum
but
depth, at
maybe it
least
was just
in this
too close
area.
together.
Once you get past about 1,000 meters, we don't see these bioherms at all. And we actually see a turnover from Lophelia and Analopsamia to Selenosmilia, which is a closely related
I think I'm starting to see
colonial
some stalk
scleractinian
sponges
coral.
in the distance.
And then
Okay.
Good turn your
we
octopus
also
off if you like
see
'em on.
Yep.
that these
them
bioherms
off.
don't form anymore once you get past Cape Hatteras, at least to my knowledge. We do find Lophelia, but it grows not in bioherms like this, but it grows down from ledges, so it's basically underneath overhangs. And it was missed by scientists for a long time. It was a gap in their distribution in the mid-Atlantic region, and it wasn't that it wasn't there, it was just that we couldn't find it very easily because you had to kind of peek underneath the ledges. So it wasn't until we visited canyons with remotely operated vehicles and were able to
I see that.
explore underneath ledges, and
Trying
that
to mosey
habitat,
in here, but
ledge
I think I'm
habitat,
gonna run out
is
before I get there.
quite a
can
lot
come
of
in
different
partial
species
here.
actually use that
It's about as
habitat.
much tether as I have, but we can check it out floating.
Oh, and this is an interesting sponge. Looks like it's on a stalk of spicules.
It's got a little red center on the top.
Well, that's one way to get up into the current.
That's
Just
the
build a
creature.
base of spicules.
Yeah. The one dive that we did that was a lot of sediment. There were many
I'll be able to get
stalk
closer and get
sponges.
a closer look once we get Sirius in a little bit.
Yeah, the boat's on the move, looks like.
And it does have a little kind of darker colored center. Looks like a bouquet.
It's incredible how strong the base must be.
Yeah.
guess the base isn't solely spicules. It's part of the sponge itself as well.
It's kind of a light pull all these off.
And Chris Kelly has a couple of ideas about what this might be. And has a Lopophytus attachment.
Just tilt to fill the screen.
And that rules out a lot of species. But it might belong to the Hyalonementidae. I hope I didn't murder that, but
you notice I'm not trying.
Very good. You didn't murder that at all.
Oh, good.
These are really cool because the stalk are actually spicules made out
Getting
of
a
silica.
lot of vibration in the
Wow.
camera, huh?
Anchoring the sponge.
Yeah.
And it's a fairly strong current, so it's anchoring the sponge into this
A lot
rubble
of
and
thrusting.
sediment. Chris Kelly is the real expert on these hexactinellids, and Scott France also has a fair amount of experience.
Okay.
I
There's a cool looking
believe this is a hyalo
glass sponge over here too. We'll go nosy that way.
You pronounced very well.
Great. Thanks, Shirley. What a beautiful sponge.
And that's the first time we've seen one of those.
And these are they're very I guess compressible. I was going to say spongy, compressible. And on this one though, it's not. We can kind of like distinguish compressible from ones that are squishy and are not resilient, and others that'll
If I do another
go
20.
back to their original shape. This one,
Copy
when you
that.
collect it,
330 this time, I think.
bring it out of the water,
Do you guys want a
that
closer look at this
beautiful
one?
bulb or kind of
Do we
tulip
know what it
shape.
is?
No, I think
We
we're
still have
body,
2 meters on
it
that last
completely
one.
collapses, and
Yeah,
it's
keep
a lot
going
like
then.
a cotton ball, a gigantic cotton ball. So if you take it out of the water, if you can imagine a cotton ball totally soaking wet, take it
Same
out of the
bearing,
water,
you
and
said?
it's just going
Let's
to
go
collapse.
330.
And that's what happens
Okay,
to
so
these
a little bit
sponges
more
when you
report.
pull them out of the water.
We'll come over to
Wow, that's interesting.
You can see the underside of that sponge
Copy that.
eventually.
we were looking at before. Just do partial on that. As we fly by.
Oh, this is the third of this type of sponge we've seen.
So without its dust or something.
I believe at the top of it has
And a
a
bunch of brittle
sea
star
cucumber.
arms, the little pink things you see on the
Oh yeah.
coral skeleton in front of the sponge.
Might just be some seagrass too. It's hard to tell.
Oh, and it has a little
hard to
brittle
tell.
star on top of the sponge on the left.
Climbing to the top. That's great.
There's a more encrusting sponge on the
Too much.
left, and maybe a little cup coral. I couldn't tell if that was a cup coral or an anemone.
Looks like a scleractinian. It's hard to tell.
ROV nav bridge move complete.
Copy bridge. Are you guys ready for the next move?
We can, yeah.
I think that looks more like a scleractinian cup coral than
Sorry,
it does an
Roland. Am I
anemone.
coming in or out?
Okay, 20
Narrow
meters.
at the
Slowly
base.
drifting
Yeah.
forward.
330,
Fanning out.
speed decimal 2.
It's hard to tell from my manip cams right now.
And these cup corals represent a lot of the diversity of deep sea corals. There are about half of the species of corals in general occur in the deep sea. And of the scleractinians, the cup corals are the most diverse at
Okay,
depth.
getting rosy on,
They're
I
also difficult to tell
Wanted them
apart
to comment on those
unless
two
you actually
arms,
collect
the
them
feathers.
and remove the polyps, so that you can see the calyx that the polyp sits
ROV
in
nav bridge
and
move initiated.
count and look at the shape of all the structures
Copy
inside.
bridge. Move initiated.
Trying to
We
get it
got
in view
some
here.
couple little stalked crinoids, baby crinoids,
And in the distance,
And
you
a-
see a tube worm.
the feather worm.
I mean, a feather worm.
yeah,
He has a tube, and you can tell that the tube is partially solidified by gluing onto the tube the
Stream
available
below.
sediment. And you can see that's a great view to show you what the sediment is composed of. Very large fossil
yeah, I
shell
see it.
remains, including a lot of pteropods.
Okay.
And our colleague Stephanie Bush is smiling. That's her favorite group of organisms. She's sitting here with us. Stephanie, you want to introduce yourself?
Hi, my name is Stephanie Bush, and I'm sitting in the control room with Leslie and Cheryl here, just admiring some of these benthic organisms. I work at the Smithsonian Institution National Museum of Natural History, and I do study.
This one?
I study pteropods, the living ones in the water column, but yeah, once they die, their shells sink down. So in all the dives
But you
this
can still
week,
wave it.
we've been seeing quite a few of them on the bottom.
Fernando's making fun of me. It's okay. Looks like we're moving.
And Les Watling pointed out that the temperature is actually fairly high here for this depth, and I agree. The temperature is looking at about 17 degrees. I'm not sure if what we're seeing is the water temperature there.
Is that free or what is going on up there?
Up where?
I think what you're seeing is back at D2.
Yeah.
You're seeing the tether, and then you're also seeing
Video
the tether
swapping out.
way back.
Okay. Thanks, Roland.
Okay.
Yeah, that's not a small line. That's the tether further away.
All right.
And this landscape is now what we've been seeing. This is our second day of sort of this dominant view with undulating portions of the larger mound. Dominated by the rubble of the coral, the dead stalks of coral that have broken down. And in between the coral, the coarser grained fossil-rich sediments that are able to deposit in the strength of the flow of the Gulf Stream.
And we just asked Nav, and the temperature down here is actually 8.5 Celsius. So that temperature that I was looking at was something within the vehicle, not the actual temperature of the water. So that's more within the range that we would expect at this
I don't know
depth.
what's going on with their SCS, actually.
It's another of those
Extra screen in
unusual
front of it.
sponges and the cousin it sponges. I guess I'm dating myself if I say cousin it.
Is that a piece of marine debris?
Not sure yet. We're getting a little closer to
It's
find out.
looking
I
a little
think.
angular.
You want to come in partial, see what
Might
this
well
is?
be.
Just see if it's
ROV nav, this is the bridge.
garbage
Our
or something.
position
Yeah.
move is
Yep.
complete, and I would like to bring the heading
Piece
2
of
degrees to starboard.
Paper or plastic?
Thank you.
Yeah.
Probably plastic. Is that a
So,
glove,
Levi,
maybe?
They're gonna bring heading to starboard 2 degrees.
Okay.
After that, you wanna move?
Yeah.
Okay. Same 330.
Yeah.
Copy.
Getting a closer look
Probably
at
do
the
30 meters.
scleractinian
Copy that.
rubble here, it looks like an Alapsamia profunda to me so far,
Serious.
which is the coral that we
Who's
saw
the
making
heavy?
a framework, especially on the
Yeah.
slightly deeper mounds yesterday.
The motors were commanding? Or
Well, actually, this
Didn't
is
see
now.
one of these yet today. Come in partial video before he spooks. Or she.
We have a rat tail, Nizumia. And I don't know if that sponge behind it is flipped over or if that's the way it's supposed to grow.
I wonder if it's that same sponge, but that we saw earlier. But it's built a mound beneath it.
All right. I'll go and take a closer look at that sponge.
Oh no, it looks different.
Yeah, it looks different to me.
Yeah, definitely. the one to the right is more like what I was talking about.
This is very strange looking, huh?
Little volcanoes.
And ROV nav, this is Bridge. We are steadied up on a heading of 197. Over. Absolutely. Okay,
and even Chris
meters,
Kelly, our sponge expert, has no idea what's going on
decimal
with this sponge. We're not the only ones, Leslie.
Just peeking down a little more. Trying to get sort of more of a top view. See
Go ahead, Nev.
Their monitor up
See
there.
a shrimp just beyond.
So with that,
ROV now,
Shirley,
bridge,
with that
move
sponge,
initiated.
it looks like there's a significant amount of kind of like dead
Copy
area.
bridge, move
That
initiated.
and I agree with Chris, it may be a rosellid, because we have seen really big rosellids off of this area in the past. That's a particular type of hexactinellid or glass sponge. And what we're seeing at the base of that, what looks like a volcano, are like the spicules that seem to be trapping sediment. The brighter white patches are live, the sponge is alive at that brighter white area. But where it looks like sediment, that's what it is. It looks like sediment's being agglutinated or trapped. I shouldn't say agglutinated, that's not the right word, but trapped in those spicules.
Okay, video.
Wow.
Ready?
Great. Thanks, Shirley. And Chris Kelly said maybe ferronematids?
Yeah, hard to tell, Chris.
Pilot, we'd like to get a zoom in on some of the sediment as
Okay.
well.
Yeah, of
I think
course.
we'll see a lot of shell material, but also we can get a close-up of this rubble that is very defining of the area.
I can also drag a toe a little if you're curious what's underneath. Come in on this section of sediment.
Yeah. Oh, nice little pencil urchin.
Wow, it's a very baby pencil urchin. I think I see pteropod shells.
I was waiting for you to point that out.
Sorry, I'm not looking the whole time.
Sorry, they're littering this area much more
The
so
one in
than
the
we've
upper
seen.
left looks like maybe Diacrya? It's hard to tell. Well, now it's lower left. Or it could be Cavalinia inflexa.
No, I think that's great, Pilot. Thanks.
Yep.
We may want to return back to that strange sponge we were just looking, if we haven't drifted too far from it.
No, we haven't drifted
There's
very
a
far.
possibility of collecting it.
I'll back up.
There were two of them there.
Bridge,
We
does
could
that-
possibly collect the smaller individual, and
Bridge
since
over.
it seemed to be stumping everybody, it might be
12
worth
meters.
collecting and getting more information on.
Okay, stopping.
Could you leave it wide for a second, video, just as we hunt for these odd looking? There they are.
Alright, co-pilot, ship stopped.
Copy that. I'm just lining up my heading with uphill.
ROV nav bridge, all stopped.
Copy bridge.
So we've put it in reverse for a little bit here, and we're coming back to the sponges, those kind of pyramid-looking sponges that we were just looking at. Since those were unusual and there was no consensus among the scientists in our chat room here, we were thinking about maybe making a collection of one out of the two of these so we could take a closer look and possibly get a genus and species on these.
Jeff, just to look uphill again.
Okay.
This is Shirley. I think that's a really good idea because this is unusual. We've been going back and forth here in our lab as well, trying to figure out what that is.
Okay.
I think that's a good idea.
Okay, great, Shirley. Would it be
pretty
okay
shallow.
to
We're not
collect
gonna-
the smaller one? Oh, look, another
swimming. No, wait, is that?
Is that no fluoride or?
that
Oh, that's a
Fernando? Is yours
percentage,
heading
isn't
always
it?
due north
I
on high
can't
tide?
tell.
Yeah, it's
Almost looks like a crinoid, but I don't think it has enough.
Was it working yesterday?
But the tracking is pretty good.
Yeah.
Can we get a quick zoom right in the middle?
I think it was working.
Oh, you're settling in.
Okay.
Sorry, pilot.
That's okay. Once I get settled, I think you'll be able
Okay.
to zoom in.
Hopefully you won't swim away. I think he'll be settled for a bit.
Yeah, I'm receiving.
Yeah, come on in, video.
The USBL bearing of. Oh no, hold on.
Five-armed crinoid.
Yeah, should be
That's a
separate
crinoid.
windows for heading, right?
Wow, it
is
is
it
a
really
crinoid.
swimming or just caught up in the current?
They swim. We've seen
It
it.
looked like
You
they were
have seen
swimming.
it?
Yeah, we've seen them swim.
I've never seen a crinoid swim. Wow, a brittle star and a crinoid swimming.
Look it down at you.
News to me.
80.
Sorry, my left toe's in and the current's coming from the left, so this dust is probably not going to clear.
And if
Get a
the
better
viewing
image.
audience was not with us on our first dive, definitely check out the video highlights of our swimming brittle star. Brittle star breaststroke.
That was amazing.
Hydraulics are ready.
Okay. Great, thank you. Tilting down to get the subject in view.
And to the right of middle is
Spun
one
up,
of
you
those
said?
little
Yeah.
mound ones
Great.
that we've been seeing all over, and
This
just
live.
at the edge of the upper right of the screen also.
Oh, yeah, there's two of them there. And Iris Sampaio is commenting that this is
All right.
quite similar to Pharonema carpenteri from the Northeast Atlantic.
Just give it a little prod real quick just to see the squishiness. It's pretty squishy. It's pretty small, huh? Just comparing my jaw with. It'll be tricky, but I think we can get it. What if the vehicle drifts away? That's no good.
So Cheryl, you asked earlier if we could collect the smaller one, and yes. And I have a very significant video delay. I'm thinking about going over to our Exploration Command Center so I can get a faster video. But yeah, the smaller one's fine.
Okay, great. Thank you, Shirley. We're going to attempt to collect the smaller one of these two individuals, and we're probably going to have to dig underneath to get the whole thing.
Would the scoop work on that?
Maybe.
Would you like me to try the scoop?
Yeah, pilot, that might be a good idea. It might wind up not squishing the sponge as much.
Okay.
On the wing in.
Yeah, Shirley, we'll try for the scoop.
Jeff?
Yep. Good.
Tell Levi I'm sorry for being a backseat driver.
No, that you would know how to collect these much better than we, so that's great information.
And I'm all for a scoop sample because we'll also get some sediment.
And maybe that sediment will have pteropod shells.
And foraminifera.
Agglutinated foraminifera. Something for everybody. And, of course, coral rubble.
That's a given.